I am Snigdha Bharadwaj, Assistant Professor, ITS College of Pharmacy. Today we have lecture on Biopharmaceutics and Pharmacokinetics basic introduction and basic definitions. So, let us get started. So, first of all, I would like to give you some information about the basic introduction of your pharmacokinetics. So, it is derived from ancient Greek pharmakon, that means drug, and kinetikos, moving, putting in motion. That means pharmacokinetics is the study, basically we observed how the drug is going through what type of processes, for example, absorption, distribution, metabolism, and excretion. So, it is a branch of pharmacology dedicated to determining the fate of substances, or we can say drug substances administered externally to a living organism. So, we just observe drug performances, basically the different processes affecting the drug performance within the body itself. So, this study involves in pharmacokinetics. The substances of interest or active drug include pharmaceutical agents, hormones, nutrients and toxins. So, basically we have variety of drug substance which we observe in pharmacokinetics within the body. So, the phases are, first is, as you can see in the picture, first is biopharmaceutics, and then pharmacokinetics, and then pharmacodynamics. So, all the three terms are interlinked to, with each other, but are different in their functional manner. Means biopharmaceutics, the patient related factors, the physicochemical factors of the drug, and the diseased and the different body parameters are well studied in biopharmaceutics part. Whereas, in pharmacokinetics we used to correlate the drug effect with the body parameters as well as the physicochemical parameters of the drug. So, it is a link between the pharmacodynamics and the biopharmaceutics. Pharmacodynamics basically is the study of the drug effect to the body. That means, how it is affecting, how it is giving different kind of activity within the body, right. Pharmacodynamics, what a drug does to the body, okay. And pharmacokinetics, what body does to the drug, that means various processes in body affecting drug performance, like I have said earlier, absorption, distribution, metabolism and excretion. These all are comes under the pharmacokinetic study. So, as you can see, I have mentioned one example, tablet. If we talk about the GIT example, so oral administration, tablet is given orally, and first it is going down through oral administration to GIT, and then it will absorbed across the GIT lumen and will go to the blood or systemic circulation, and then it will be distributed in the body cells, that is Q receptors, tissue and all. The distribution will occur, and then metabolism will occur, followed by excretion. So, this is the basic pattern in we observed in pharmacokinetics. So, we can say that this is the basic pattern through which drug is first absorbed, then distributed among the tissues, body cells and tissues, and then metabolized into other form, and then eliminated or excreted. So, pharmacokinetics and pharmacodynamics. So, both I have given you detail, but once again you see that the figure represent a dose of any kind of pharmaceutical dosage form or any kind of pharmaceutical formulation like tablet, capsule, suspension, syrups and all. So, first you correlate this pharmacokinetics. First, the tablet or dosage form is going into your first is it is administering to the body part, and then it is well absorbed and distributed, metabolized and excreted. So, same here, when it is absorbing into the blood, so that means the plasma concentration of the drug will be high. As the higher the absorption, the plasma concentration of the drug will be higher. Okay? And then when the plasma concentration of the drug is high, that means it will reach to the site of action. We can say with the previous picture, receptor tissue distribution basically. So, site of action, it will reach to the site of action and affect the body receptors, and then you will get the required or desired effects. Like if you see, the paracetamol is analgesic and antipyretic. After reaching the site of action, it will give you analgesic or antipyretic effect, pain relief. Okay? Then you can say that the drug is affecting once it reaches to plasma concentration, or it reaching to plasma or blood, then it will show it will start showing effects. So, first we have given dose, and then it will reach to plasma, and then concentration is high, then site of action, and then effects. So, the process of reaching that dose to the plasma is considered pharmacokinetic studies, whereas when it will from the site of action and showing the effects, desired activity. So, the study will come under pharmacodynamics. So, I hope now you are able to understand the basic difference between pharmacokinetics and pharmacodynamics. Now, move on to the next topic. This is topic means definition. This is clinical pharmacokinetics. The synonym is also applied pharmacokinetics. Basically, it is the application of pharmacokinetic principles to the safe and effective therapeutic management of drugs in an individual patient. So, basically, when we talk about clinical pharmacokinetics, it is the application of pharmacokinetic principles to the safe and effective therapeutic management of drug in an individual patient. That means, we are observing its efficacy and safety, whose efficacy for drug effectiveness and safety in individual patient clinically. Okay. So, how we can get the desired pharmacokinetic profile of a drug in an individual patient. So, primary goal of clinical pharmacokinetics include enhance efficacy and decrease toxicity of a patient drug therapy. Yes. For example, we are observing during formulation development. So, from the pharmacokinetic study, we can establish that the drug is absorbing from this type of barriers, like for if we talk about tablets, solid dosage form. So, it is getting absorbed through GIT lumen, that is the GIT lumen is known as barriers. So, various mucosal layers or barriers or skin barriers are rate limiting, or we can say steps which can affect the drug absorption. So, analyzing all these absorption, distribution, metabolism and excretion parameters to get the maximum efficacy of drug and to reduce the toxicity is included in clinical pharmacokinetics. When we observe, when we the primary goal of studying clinical pharmacokinetics to observe the parameter for higher safety and efficacy of the drug. Now, pharmacokinetic processes. So, first, as I have told you, there are basic processes: absorption, distribution, metabolism and excretion. So, first we will discuss about absorption. So, in absorption, many terminologies are there like bioavailability. So, bioavailability is the rate and extent of the drug reaching to systemic circulation is known as bioavailability. The percentage of the administered dose that reaches the systemic circulation, how much drug reaching to that particular blood or systemic circulation. As I have told you that you can see that you are giving some dose to a patient. Now, how much of that particular dose is reaching to a patient or reaching to a patient's blood, or we can say plasma concentration may vary. If you are giving 100 mg of the drug, maybe the drug dose reaching to a plasma or blood systemic circulation may be 50% or 60% of that particular drug. So, that is the bioavailability of that particular drug. So, here this bioavailability refers because the amount reaching to your blood circulation is the amount which shows effect to the patient. Like for example, if I am giving 200 mg of blood of drug to a patient, administering 200 mg of a drug to a patient, and the bioavailability is just 50%. So, I can say that only 100 mg dose of the drug is reaching to a blood circulation at that point of time, right. So, bioavailability will become 50% of that particular drug. So, absorption rate, after this bioavailability, absorption rate, the speed of input into the systemic circulation, at which speed, for example, in 30 minutes from 0 time to from 0 and after 30 minutes, how much drug is getting absorbed into the plasma or reaching to systemic circulation is known as absorption rate or rate of absorption. It is denoted by Ka. So, basically in which particular time the drug is reaching to, how much drug is reaching to systemic circulation is known as absorption rate or rate of absorption. Distribution. Next, the process next to that absorption is distribution. After coming or after reaching to systemic circulation, now drug will start spreading or distributing among the body tissues, right. So, in this volume of distribution, this is the important parameter to identify the drug distribution pattern among several receptors. So, the apparent volume into which the drug distributes within the body, right. So, we considered body as having multiple compartments or single compartments. So, from that study we can identify volume of distribution, that how much volume or how much drug is diffusing in how much of volume in the body compartment. So, the basically that particular parameter covers that aspects. Protein binding, of course, if protein binding is not there, then the drug effect will not occur. So, protein binding is the percentage of drug binds to plasma protein is known as. So, we have albumins, we have different kind of proteins present in our blood. So, after reaching systemic circulation, the drug will distribute it and bind to the particular protein to show its effect in the body. Now, pharmacokinetic third process is elimination. So, elimination, basically in elimination we have clearance, because in metabolism, yes, after this metabolism is also important process, absorption, distribution, but metabolism, how drug is metabolized or converting into the inactive form from the active one. So, biotransformation, various reactions are there, and it is mostly occurring in liver. Hepatic metabolism are usually seen for most of the drugs. So, after that elimination, clearance. So, first is clearance, the volume of drug cleared per unit time. So, when we talk about that drug is eliminated from the body, we say that means the clearance, right. So, volume of drug cleared per unit time. Elimination rate constant, that means the proportion of drug in the body eliminated per unit time. The proportion of the drug, the amount of the drug, we say that if 50% drug has been reached to systemic circulation, how much is getting eliminated through the system? Basically, the proportion of drug in the body eliminated per unit time, right. The total amount, from the total amount we observe the how much proportion is getting eliminated through the system. Elimination half life, that is also known as T half. The time taken for the concentration to fall to half the previous value. That means, overall the drug amount, if getting reduced by its half, is known as T half. In which time, in how much time it is getting reduced to its 50%, that time is known as half life of drug, right. So, these are basic terminologies which we use in our upcoming sessions. So, basic other again the pharmacokinetics is defined as the kinetics of drug absorption, distribution, metabolism and excretion. So, basically kinetics, we use word kinetics. So, how it is systematically approaching, right. First absorption, second distribution, third metabolism and fourth excretion. So, it is also known as KADME, KADME, right, and their relationship with pharmacological, therapeutic and toxicological responses in humans. Yes, of course, we observe these processes, but we also observe the connection of drug to these processes, like how drug is absorbing, how drug is eliminating through the systems and their relationship with each other. That means, if the processes are well studied, then we can have the profile of drug toxicology, drug safety, drug efficacy as well. So, we can correlate the drug effect by analyzing these different kinetics. Absorption is defined as a process of movement of unchanged drug from the site of administration, unchanged, unaffected drug. If the drug once degraded, that means there is no use of absorption of that particular drug in a degraded form, right. It will not show any effect which is required to the patient, right. So, absorption is a process of movement of unchanged drug. As I told you that if a patient takes tablet, that means the tablet first go to the GIT lumen and then absorbed, first disintegrate, then dissolute and then absorption will take place. So, it is a movement of unchanged drug from the site of administration to systemic circulation or to the site of measurement, that is plasma. Distribution is reversible transfer of a drug between blood and the extravascular fluids and tissue or peripheral tissues, we can say. The blood is considered to be a main compartment of the body system, and the other tissues and distribution centers will be considered as the peripheral compartments of the body. So, how the drug is distributing, first it is coming to the blood component and then it is distributing to the body cells and tissues. Elimination. Elimination is a major process for removal of a drug from the body and termination of its action. Once the drug is eliminated through from the system, that means the action of drug will not occur. It is defined as the irreversible loss of drug from the body. Elimination occurs by two processes, with biotransformation and excretion. Biotransformation means that the drug is biologically converted into another form which is inactive, right, and excretion, renal excretion we can say. Metabolism, that is biotransformation basically. So, under elimination, metabolism is a part. Yes, hepatic metabolism, majority of metabolism processes for drug substances occur in liver. Yeah. So, in liver is, we can say, the center of biotransforming the active drug into inactive form, right. And then excretion. Excretion is defined as a process whereby drugs or other or their metabolites are irreversibly transferred from internal to external environment. Okay, right? The excretion means the body is throwing, or we can say the drug is eliminating from the system and going outside from the body in terms of feces, in terms of renal excretion. Okay. So, basically we have these processes under pharmacokinetics. Now, pharmacokinetic studies have two aspects. First is theoretical aspect. So, this aspect involves development of pharmacokinetic models to predict drug deposition after its administration. Theoretical aspect just means we consider various processes, and theoretically we assume that this will be a fate of drug when given orally or any from any route of administration. So, theoretically we analyze the things, or based on these different processes named absorption, distribution, metabolism and excretion. So, statistical methods are commonly applied. So, we use some sort of mathematical formulas to identify drugs fate in the body. So, basically it is the initial or primarily effort we use to set for drug performance in the body, and then we clinically tested the drug, how it is performing in the body, how the pharmacokinetics processes are studied, and whether it is correlating with the hypothesis set or not. An experimental aspect involves development of biological sampling techniques. Yes, of course, using the mathematical calculations, we generally develop experimental models, and then we perform the drug we perform the experiments to check the drug behavior in the body, and how the processes kinetics affecting the drug absorption or drug performance within the body, right. So, or we can say it in vitro or in vivo evaluation, right. So, all these parameters are used to analyze these pharmacokinetic behavior of a drug. Methods, analytical methods from measurement of drug and metabolites, concentration in biological samples, and data collection and evaluation. So, we match both the things from theoretical aspects and the experimental aspects, and then we can say that the predicted assumptions are matching with the experimental results. Several relevant terms are clinical pharmacokinetics is defined as the application of pharmacokinetic principles in the safe and effective management of individual patient, right. Population pharmacokinetic, the clinical pharmacokinetic means identifying the safe and effective management of the therapy in individual patient, whereas if we talk about population pharmacokinetic, it is defined as the study of pharmacokinetic differences of drug in various population. Basically, in this particular population pharmacokinetics, physiological variation plays an important role, right. So, one individual may be different from the other one based on their physiological differences. So, if we say that a particular drug is affecting so much in one individual, it may be less effective in other individual due to its physiological variations. So, the study of identifying this physiological variation comes in population pharmacokinetics. Toxicokinetics is defined as the application of pharmacokinetic principles to the design, conduct and interpretation of drug safety evaluation studies. To just observe the toxic profile and the safety profile of drug which is administering to the body for its effective use, all the observation we identify under toxicokinetics. Pharmacodynamic parameters are minimum effective concentration. Basically, minimum effective concentration is the that concentration in plasma required to produce the therapeutic effect. It reflects the minimum concentration of drug at a receptor site to demonstrate the desired pharmacological response. That means, we say that it is the minimum dose required to show effect. Maximum safe concentration, minimum toxic concentration is referred as maximum safe concentration, and it is the concentration of drug in plasma above which adverse or unwanted effect will occur. So, this is the important concern, that the drug should not reach above it MSC. Onset of action, it occurs when the plasma drug concentration just exceed the required MEC, minimum effective concentration. Onset time, that means the time at which the drug start producing pharmacological response, and then duration of action means the time period for which the plasma concentration of drug remains above the MEC. That means, the effective period of drug in which the drug is showing its effect to the body, right. So, that is known as duration of action. Intensity, how much the drug is giving its effect, the peak response we can say. Therapeutic range, the drug concentration between MEC and MSC represent the therapeutic range, the safe range of drug dose, and it is also known as therapeutic window. Therapeutic index is the index, it is a ratio of dose required to produce required effect or lethal effect to dose required to produce desired effects. So, it is a ratio or difference between these. So, this is known as the therapeutic index. These are references, basic terminologies we have covered in first introduction to pharmacokinetics. I hope you understand all these basic terminologies. We will be continuing these lectures in upcoming sessions. Thank you for your patience listening.